Hi, everyone. Thanks for joining. This is Dr. Sette. He's going to be giving his talk. He's a professor and member of the La Jolla Institute of Infectious Disease and Vaccine Center. His research focuses on developing disease intervention strategies against cancer, allergy, autoimmunity, and infectious diseases. He researches COVID-19, dengue, Zika, malaria, tuberculosis, pertussis, and shingles, and he oversees the design and the curation of the efforts of the National Immune Epitope Database, the IEDB. So, Dr. Sette, if you'd like to take over now.
Sure. Okay, let me see. Okay, this one is better. So let me now share my screen so that I can then project the PowerPoint. Do you guys see the screen?
Yeah, we can see it.
Okay, good. So as the title says, what I'm going to talk to you about today is our studies in the SARS CoV2 specific adaptive responses. So first of all, how deep is your immunological wisdom? Can you help me out here? Let me know what level of immunological training do you guys have?
Some of us have been in immunology labs before, but I would say roughly a lot of the people here are freshmen and haven't really taken many immunology based courses. Maybe just I would say some are taking our immunology courses, but I would try to teach it at a level where I think everyone would be able to understand it.
Okay, so
yeah,
I will give it a shot, and you guys let me know if I'm going too simple or too complicated. Feel free to jump in. So first of all, this is really a collaboration between my lab and the lab of Shane Crotty. Daniela Weisskopf is an assistant professor at the La Jolla Institute, and Alba Grifoni is an instructor, and they contributed very, very strongly to the research I'm going to show you. Now, our studies in SARS-CoV-2 started actually fairly early, late January. And the reason why that is that, as you mentioned, I run the immune epitope database, which catalogs all sort of immune reactions. We talk frequently with our colleagues at the NIAID, and so they flagged us on this new virus that was coming up. Of course, back then there was no information regarding, this is super annoying, keeps switching. No information regarding what were the actual targets recognized by the immune system, the human immune system in this new virus. We and others noted that this novel virus, but then was SARS-CoV-2, was really similar to the old SARS, if you wish, the one that circulated 17 years ago. So we saw a strong similarity both at the level of sequence homology, at the protein and genetic level, and also in terms of modeling the structure. We realized that these two viruses were very, very similar. So we reason, even though there is no data about what the immune system recognized in SARS-CoV-2, there was a lot of data about what does the immune system recognize in SARS. And so we extracted the information from the immune epitope database, and then we went ahead and mapped this epitopes into the sequence of SARS-CoV-2, and actually found that they were very conserved in the two viruses. So we predicted that these regions will also be recognized by the immune system in SARS CoV2. In fact, we predicted the fragments of the virus that were likely to be recognized by CD4 T cells and CD8 T cells, the helper cells and the killer cells. And so this was published early on. Then we move ahead and actually did experimental work. So at that point in time, if there was essentially no positive case of SARS-CoV-2 in the United States, there was in Europe, and then of course later on we had cases in the United States. And our approach has been to define what are the fragments of the virus that are recognized by the immune system, as illustrated here in this cartoon. So this is the structure of SARS-CoV-2, and we basically take it apart down to its pieces, and this is what it looks like in reality. We synthesize hundreds and thousands of different peptides that encompass the whole virus, and then we test them for reactivity with peripheral blood mononuclear cells, lymphocytes, or PBMCs, from different individuals. So this approach has been validated in many different systems. As you were saying in the introduction, we've been studying a variety of different viruses: dengue, Zika, you name it, flu, other RSV, and so forth, bacteria like tuberculosis, pertussis, allergens such as pollen, mouse, house dust mite, and so forth. And so we knew how to do this. And we did synthesize three main, what we call megapools. So pools of several hundred different peptides. And has to be made in a certain way with sequential lyophilization, but those details are not relevant here. But we did these three different pools. One, we predicted peptides recognized by CD4 T cells, two different pools, about 600 fragments. Then we synthesized overlapping peptides spanning the entire sequence of the S protein, because everybody was really already early on very focused on this spike protein, which is the main target of neutralizing antibodies. And then we synthesized also about 220 peptides predicted to be recognized by CD4 T cells from the remainder of the genome. So the spike we could test for both CD4 and CD8, and these two were focused on the remainder of the genome for CD4 and CD8 T cells recognition respectively. So at this point in time, we wanted to then ask the question, what does the immune system, what does the immune response look like? At that point in time, actually, there was a lot of confusion whether the immune system could mount an effective response. There was concern that there was a cytokine storm, somehow maybe T cells were involved in that. So it was very important to actually have the reagents to allow to measure the immune response. In other words, if you can draw a picture, if you can take a picture of a situation, then you can argue whether you like the picture or not. But if nobody can take a picture of a situation, there is no discussion of whether this is good or bad. So we went ahead and started to ask what kind of immunity is there to COVID. Of course, there are three main arms of the adaptive immune response. The antibodies that are made for B cells, and those are important. Almost all licensed human vaccines work by creating a titer of antibodies that is so high they can neutralize all or most of the microbe that is trying to infect the body. Then there are the killer T cells, or CD8 T cells. This is very important, this type of cells. They are almost the opposite conceptually of the antibodies, because once a virus is inside the cell, the antibodies cannot reach it. So you need a killer T cell, a cell that can actually detect an infected cell, which is what CTL do, and kill it. Then there are helper T cells. Those are very important. They support killer T cells, but are particularly crucial for the development of antibody responses. Without helper T cells, you really do not have a good antibody response. You need helper T cells to mature the antibody response, to make it strong, fast, and well neutralizing. Also, helper T cells have some degree of protective effect in SARS mouse models. Now, we decided to target as our first study convalescent individuals that were recovering from a mild COVID case and actually did not require hospitalization. And why was that? That was by design, because we wanted to see what does the immune system do, what does the immune response look like when there is a successful immune response, when the immune system can get rid of a virus without having to be hospitalized. And as a control, we ran samples from individuals that had donated blood in 2015 to 2018, so before SARS was around. So we were sure that these people had never seen SARS. And good news, we detected good responses, both in the cases of looking at CD4 T cells or CD8 T cells. The particular assay we use is a very fast ex vivo activation induced marker assays. So we do not need to culture T cells in vitro that could alter their phenotype. Importantly, this good T cell response was also well coordinated with antibody responses. Here you see the coordination of the immunoglobulin IgG against the receptor binding region of the virus with CD4, and whatever you're looking at, spike or non-spike, here's the total CD4 against the total CD8 response. So overall, good news, we see a good immune response to SARS-CoV-2. 100% of individuals, we could detect a helper T cell response and an IgG response. IgA, which are the type of immunoglobulin that are associated with mucosal secretions, so important in that respect, and also very high frequency of CD8 killer cells. So this also provides us with a kind of a landmark or measuring post of what we would like, perhaps optimally, a vaccine response to look like. Now, next, what we also did, we made overlapping peptides spanning the entire sequence of the virus proteome, and we measured against CD4 and CD8 response. So why is that important? First of all, because we were interested to see spike, and you see that spike, of course, induces a good immune response. We knew that from the previous experiments, and therefore it's a reasonable target for vaccination. Spike, if you wish, has it all. Has both the capacity of inducing a neutralizing antibody response, but also of inducing a CD4 and CD8 response. You don't want to use an antigen that is only capable of inducing one type of immune response and not the other. At the same time, you see that there are several other antigens that are also recognized prominently. And so this could be considered for additional vaccine designs where you desire to bring additional firepower to the table. In fact, we've been studying this this issue in much more detail. We now have studied almost 200 people, and we map for each individual what particular protein in the virus is recognized. And so this established patterns of immunodominance for both CD4 or CD8, and also tells us for each particular donor which particular antigen is good. Different donors have different patterns of immunodominance, as you can see. And so then we can now map very accurately for each donor which particular fragments are recognized. We're also performing experiments in which we are following, this is just a representative set of data. Bottom line here is you see days from symptom onset. We now follow people for 6 months or so, and we see that the responses against the virus, both in antibody, CD4 and CD8, are durable. So in conclusion for this first part, these mega pools that we generated based on bioinformatic predictions or synthesis of overlapping peptides really allowed to detect robust responses in uncomplicated cases. So very important in terms of the prospects for developing a vaccine. This should be feasible. And obviously, we are waiting to hear the results of the experiments that are the vaccine trials that are ongoing. We defined these targets recognized in this patient, and we see that S is a good antigen, but also identify additional targets, duration of responses, and this large-scale effort to identify different epitopes. This is in progress. Now, let's turn for a second to a study of responses in acute phase. And now we are interested in looking at the response in the acute phase and specifically asking, is there a difference? What's the difference? Why some people have a fairly uncomplicated viral infection while other people really have a terrible time and require being in the ICU or worse, they might die. So we did, we have one paper that came out early on in collaboration with a group of Robert DeVries and Richter Swart. And I'm not going to show you the data in detail, but we saw both CD4 and CD8 again, and already present at time of the admission and increased over time. So really no evidence that the T cells were doing anything bad in the acute phase. And it was a particular type of cytokine, which is what we expected, a Th1 interferon gamma producing predominant type of helper T cells. Shane Crotty has then been studying this issue in more detail in collaboration with our group, and this result just came out in Cell last month, I believe. So first of all, we can detect antibody responses in all individuals. And you can see that in general, by different types of IgA or IgG, spike, neutralization of the virus, all these different measures, really there is no difference between the acute and convalescent phase. And there is also not really a big difference in terms of whether the case is severe or not. Now we can detect CD4 responses, and now the situation is different. You see that while everybody in the convalescent phase has a good CD4 response, in the acute phase, it's more of a smear. Some people have a good response, other people do not. So in fact, when you look at this phenomenon, in a more rather simplistic fashion, if you wish, and you ask, okay, all the people that had severe versus mild outcome of disease, how many did have a strong antibody titer or a weak antibody titer? And you see that really there is no difference in terms of the breakdown of severe versus mild when you look at high or low antibody production. Instead, if you look at the CD4 T cells, there is a very significant difference. In other words, the people that have low CD4 almost invariably have a severe disease, while the people that have high CD4 almost invariably have a mild disease. The same picture is true for CD8 killer cells, but this difference is less prominent. So in fact, we combined this data in what we called an adaptive immune response index. So we say how many people have either one or none of these responses, or have at least 2 out of the 3. And you see that now that this is the best correlation we can find, where really having a coordinated immune response with multiple arms of the immune system beating on the virus at the same time is what is associated with the best outcome. And this patient actually was quite interested because really didn't have any antibodies and had T cells and was able to clear the virus perfectly fine. So this is a more complicated way to make the same point. So here you have lots of different immune readouts, and some are linked to CD4 T cells, some are linked to CD8, some are linked to antibodies. And the darker the color means positive correlation, the darker red color means negative correlation, and the number of red or white dots indicate the level of statistical significance. So first of all, you can see that really it's a coordinated response, so that there's a lot of CD4 things that happen at the same time, or same thing for CD8 or antibody response. You can also see that there is a coordination, so that CD4 readouts also coordinate and are significantly associated with antibody readout, or CD4, CD8, and so forth. So again, seeing at this larger correlogram level, the fact that the immune responses are coordinated at the level of the acute phase of a response. Now, if you do this and you look at age, it's very interesting to see that while younger individuals have a coordinated immune response where the CD4 and CD8 remain well coordinated by and large, when you look at the older individuals, this coordination, sorry, I was doing very well. This coordination breaks down. So there is a lot less coordination in older individuals. So that one of the key features of the less effective immune response in older individuals is the lack of coordination between the immune responses. Now, the thing that we've seen correlated most dramatically with the increased age is a progressive shrinkage of the fraction of T cells that is naive and can thereby recognize new viruses. So in other words, if you want to visualize this as a take-home message in a cartoon, that as the immune system gets old, it's difficult for the this dog to learn new tricks and recognize the new virus. So at the end of the day, the summary of these studies in acute phase have several take-home points. The speed of the adaptive response is likely key. This I did not show you, but really it's important to start early and individuals that do not mount an immune response effectively for a longer period of time tend to do poorly. It's important to have a coordinated immune response. Really, we have 3 branches of the immune system for a reason. It's like an army that has Navy, Air Force, and infantry. It's good to be able to have 2 different arms that can help each other in fighting a war against the virus. And age being a major risk factor, both in terms of poor coordination and limited repertoire for naive CD4 and CD8 T cells. As you might have noticed in the graphs that I showed you earlier, where I emphasized the fact that we did see a good response in the convalescent people, you probably noticed that there was also somewhat of a response in the negative control. The negative control was not so negative. And in reality, we have detected this pre-existing reactivity in people that were not exposed to the SARS CoV2 virus. This has been confirmed in a variety of different studies. For example, in the Netherlands, the study that I showed you before, collaboration with the but also studies completely independent of us. In Germany, in the UK, in Singapore, in Italy. So really emphasizing that this must be an important, a reproducible finding, but is found from different groups all over the world. So the question is, where does this pre-existing immune reactivity come from? And the hypothesis that we and others made is that this could come from common cold coronaviruses. These are, as its name says, viruses that are responsible for common colds. So the not so evil cousin of the SARS-CoV-2, the good cousin of SARS-CoV-2, and they commonly circulate all over the world. Now, this is important because in some people, somewhere between, we saw in 50% or maybe as low as 20% in other instances. But if these individuals have some degree of pre-existing immunity, they could have potentially, the immune system could have an advantage in the sense that they could mount a faster, better immune response, and this could influence disease severity. So they will still get infected, don't get me wrong, but they would have an advantage. And the same is true in potentially in vaccination, where people that had already pre-existing immune response could respond better to the vaccine. We decided to look in this topic in more detail. These were interesting hypotheses, but hypotheses nevertheless that needed to be addressed by experimentation. And so we looked at some additional cohorts of unexposed individuals and control cohorts of COVID patients. And we could see that the unexposed individuals that we were studying, of course, we were negative for SARS-CoV-2 antibodies, but each individual was actually positive for 2 or 3 of the common cold corona, which is what you typically see. Everybody has antibodies against these viruses. We went ahead and defined a total of 142 different epitopes in these non-exposed donors. You see some epitopes are recognized in multiple donors, so are stronger epitopes, and they account for a stronger fraction of the response. So specifically, we were curious about whether the activity of these non-exposed individuals is really due to cross-reaction with common cold. And that's what we tackled in this study that was published a couple of months ago, 3 months ago, I think, in Science. So first of all, we did some sequence analysis. We asked the question, if it's true that this has something to do with reactivity common cold coronas, then you should see that the epitopes that are recognized more strongly in multiple donors have some higher degree of homology to common cold corona. And that was indeed found to be the case. Then we went ahead and did additional experiments. And so what did we do? We picked 30 or so epitopes from a spike and 30 epitopes from the remainder of the genome. And we tested them for recognition in unexposed individuals. And then also we went ahead and synthesized the corresponding peptide, the homolog peptide, in the 4 different common cold corona. So approximately, you see here, 30 times 4 is 120 peptides. We also consider additional variants. And bottom line, we saw that when we tested the analog of these sequences target by unexposed individuals, we synthesized the homolog from common cold corona, they were recognized even better than the SARS CoV2 sequences. Again, implying that what was really recognized in vivo in these donors unexposed to SARS CoV2, that these were exposed to common cold, and they had some T cells that then were able to spill over and cross-react on highly homologous sequences from SARS CoV2. In fact, we were able to show, I'm not going to go through the data in detail, but you can take my word for it, that we could show that these T cells from unexposed individuals that recognized SARS-CoV2 sequences were memory cells. So these were cells that were not originating in the short assay that we are using here, but had encountered some antigen in vivo, and they were experienced memory cells. We went ahead and further synthesized the homologs, and now at this point we made T cell lines by simulating the lymphocytes of the unexposed donors in vitro for a couple of weeks. We make these T cell lines, and this allows us to isolate specific T cells for one specific fragment and ask the question whether the common cold corona analogs are also recognized by the SARS-CoV-2 cross-reactive, allegedly, T cells. And in fact, we saw, yes, they are recognized. And you see here this particular HKU1 peptide, which is highly similar to the SARS-CoV-2, is recognized just as well. And in fact, in a number of instances, we could see that the responses to the common cold corona were even higher than the responses directed against the SARS-CoV-2 sequence. Again, suggesting that perhaps what had induced the immune response in vivo was the common cold corona variant. In additional T cell lines, we could show that this was true in multiple donors. This particular donor, 2209, recognized this P96 peptide, and it so happened that this other donor, 2086, also recognized the same peptide, so we could make T cell lines from these 2 different donors, and we found the same result, that in both cases the T cells were cross-reactive, with a slightly different pattern, of course, because they come from different individuals. We also saw, in some cases, T cells that were non-cross-reactive, epitopes are non-cross-reactive. And typically, when that happens, the sequence homology is low. And also, the T cell lines are not very good. They require a lot of antigen in order to be activated. So in our experience, this is synonymous with these T cells being low affinity, this activated by the in vitro simulation over 2 weeks. And so these are probably coming out of naive cells, not from memory cells. Also, this one allowed us to set a threshold of homology associated with cross-reactivity. So in conclusion, we answered the question whether the reactivity detected in non-exposed subject is real. Yes, it is real. We've seen it in many different cases in different continents and so forth. Can we demonstrate reactivity in non-exposed and show that this maps to a specific fragment of common cold corona? Yes. And as I was saying, the jury is still out whether this pre-existing reactivity does influence immunity, in the sense that is it true or not that individuals that have this pre-existing reactivity have, when infected, have a lower degree of disease severity. We are very eager to share our data and reagents. We publish our data, we put them in the immunoepitope database where they are freely accessible to the scientific community worldwide. And also we have, we shipped our reagents to over 100 different laboratories worldwide. With that, I would like to start drawing the overall conclusions, reminding you of the main points that we hit during our this last hour. So robust CD4 and CD8 responses in the convalescent uncomplicated cases. So this is a snapshot of what a success story looks like. The immune system can fight this virus and succeed. We define what does the immune system look like, the immune response look like, both in terms of the types and magnitude of a response, but also what are the targets. We also see that T-cell responses are durable over 6 months or so, which is very important. There has been a lot of concern that maybe immune responses to coronaviruses in general are short-lived. The study of the acute phase, in particular severe cases, suggests the speed of the adaptive response is likely key, and the coordinated adaptive immunity, in the sense of having all three arms of the immune system cooperating in fighting the virus, is a really important factor. And of course, age is a major COVID risk factor that was known, but we have provided evidence that this is really a breakdown of coordination and a decrease in, related to also a decrease in the repertoire of naive cells that are available to mount a de novo response. Finally, this cross-reactive immune memory is reproducibly detected in non-exposed subjects, and this reactivity in non-exposed subjects is shown, at least in some cases, to be straightly due to cross-reactivity with common cold viruses. Finally, the question that still remains to be addressed is whether immune reactivity can influence immunity outcome and severity of infection. This is actually Tony Fauci waving our Cell paper in a congressional hearing, which was quite a good feedback. And with that, I'd like to stop and I can take any question.
Okay, so if you have any questions, you can just raise your hand and then we'll ask to unmute.
Oh, Marissa, go ahead.
Yeah, I was wondering if you or anybody might have like any ideas, or if there's any research about what may cause like the speed of like the immune system like starting to I don't know what the right word is, like react against the COVID maybe less. What the reason for that might be?
Yes, this is an excellent question. So in the perfect world, what things are supposed to, the way things are supposed to work is that when the virus gets in, there is a first line of defense, which is
the innate immunity, right?
exactly the innate immunity. So there's actually a couple of papers, excellent papers, came out recently in Science that showed that a very high fraction of people that have very severe outcome have antibodies
towards IFN, right?
Exactly. You're very on top of things. Against innate immunity mediators such as interferon or have mutations in there. So basically what happens is that in those cases, the innate immunity doesn't control the virus. And so the virus starts to reproduce, and that kind of, we think, throws off the balance of immune response. And so the, so to speak, the machine starts late. And so, but at that point, by the time you get CD4 and CD8, the virus is already out of control. The virus titer is higher, and so forth.
So that might be like a large part of it, or a part of it.
Yes. So it's as Shane always says, that each virus has at least one good trick to fool the immune system. Otherwise, it would not be a successful pathogen. So, but yeah, I think that's likely what's going on.
I guess my question is, earlier you mentioned that older individuals are more likely to have a more acute response to COVID, but you also mentioned that having immunities to different of the like common cold COVID viruses can help you. So I'm wondering if there's because a lot of older people will likely have more of these immunities towards the less severe common cold coronaviruses. So why are they more susceptible to having acute reactions to COVID?
So, not necessarily so. So the first of all, it's the reason why people get severe COVID. It's very multifactorial. So we just discussed, for example, the defect in interferon, and there are other autoimmunity and antibodies to interferon may actually increase with age, for example. Obviously, other things such as obesity or heart problems or diabetes also tends to increase with age. So that's one thing to bear in mind. But what you touched on, it's a very, very important question that we are studying. More generally, is not really, there's not a lot of good data about the frequency of infections and specifically of T-cell responses to common cold as a function of age. In reality, kids get in the early stages, kids are not very susceptible, but then there is a stage where, you're familiar, if you wish, in your common experience, there is a period where kids are always with colds, right? And parents also always get colds from the kids coming home from daycare. So we are in the process of really looking at the common cold reactivity as a function of age. We're curious to see whether that correlates to a certain degree with the disease susceptibility. And is also true that, for example, it might correlate with different occupational stories. For example, healthcare workers, they might get exposed to a lot of common cold corona just because, you know, if you're before COVID, right? So you just take care of sick people and they come to emergency room where they have colds and they have all that kind of stuff. So it's possible that the healthcare workers may have, pediatrician, for example, may have higher common cold reactivity. That though is complicated by the fact that now the healthcare workers, of course, have higher level of SARS CoV2 exposure because that's unfortunately what they do as compared to someone that is staying at home in a lockdown. So those are things that we're trying to dissect, and but it's not necessarily a linear relationship.
Hello.
Hey, Brian.
Yeah, I was wondering that in addition to like looking for postdocs, is your lab going to be looking for undergraduates as well?
We usually don't have a lot of undergrads, but we could consider it. In general, we do have at the La Jolla Institute, I know, for example, Shane has undergrads, so it's possible. So if you or others are interested, by all means, shoot me an email.
Okay, thank you.
I had a question. So you said that you had particular epitopes which shared higher homology and with other human common coronaviruses, and that's what the root cause of the cross-reactivity is. And was there any study? Was there? Did you observe like some epitopes being more common from certain type of populations? Because we've seen that, and I'm not sure about this, but I think there is like certain populations are more prone to particular type of human common coronaviruses, and that can make them more or less susceptible to COVID-19. So is there any like correlation, I guess, between?
Yeah. Absolutely. Very interesting question. Absolutely. We want to look into that. We don't have any data, but certainly that's another interesting hypothesis, which is why, for example, if you look at Italy, for example, North Italy was hit much more severely than Central Italy. Does that have anything to do with a different pattern of circulation of the viruses? Possible, but in general, it would be very interesting to, again, compare the degree of cross-reactivity with degrees of disease incidence and disease severity in different areas of the globe.
Yeah, it's also that, you know, different HLA alleles have different frequencies in different populations, and certain populations are would have more of those alleles.
Yeah.
And those, like, I think to a large extent, the MHC binding would be a great influencing factor when it comes to epitope binding, and maybe certain HLA alleles are like we know certain HLA alleles being more like putting you at more or less risk to severity.
But it doesn't look that way. Again, reasonable hypothesis. In fact, we, as I was mentioning, we have now looked at the immune reactivity of several hundred people, and we do HLA type all of them. And so we were looking, is there anything obvious that jumps out that certain particular alleles are more frequent in people with COVID? And so far we haven't seen anything that it's worth is jumping out at you. Now, of course, you could ask in the HLA association, you could say, well, but you need to look at 10,000 people. Fair enough. However, if you need 10,000 people to look at to detect the effect, then it's likely not, it may be real, but not one of the driving forces.
All right, that's interesting. Thank you.
Hello, I had another question. It's kind of ties to what you mentioned earlier, but since COVID is so similar to like the other common cold relatives, why do you think it makes such a successful pathogen compared to its relatives?
Well, so who knows? You could speculate in a couple of different directions. One is that 10 years from now, SARS CoV2 might be the 5th common cold coronavirus. It could be that one of the reasons why it's creating such a havoc is that it's totally new. So nobody has pre-existing immunity, while everybody has pre-existing immunity to common cold. Bear in mind that young kids, for whatever reason, don't get sick with SARS-CoV-2. So you could envision a scenario where over time, the human population would develop not immunity, but a resistance, if you wish, in the sense that, like for the common cold, you still get sick, but you have been exposed multiple times, and so nobody knows what a common cold would do in a completely naive population. The other dimension is, as we were talking about a minute ago, is that this virus may have some very strong aces up its sleeve in terms of interfering with innate immunity and so forth. Finally, what's successful in general, pathogens are not supposed to kill their host. That's a very bad strategy from the point of view of a pathogen. The ideal pathogen is basically a mild parasite, if you wish, can coexist with his host, doesn't make the host very sick, allows the host to infect everybody. Like, for example, the CMV or EBV viruses that infect everybody, and it's fairly mild, or some of the influenza strains. So what usually happens is that if a virus that is used to a certain animal host and then jumps into humans, has not developed yet a balance of how to interact with humans. So it could be that also the virulence of SARS is that it's not used. Humans are not used to SARS, and SARS is not used to humans.
I had another question. I was kind of wondering if maybe you ever thought about or considered doing the sort of same study you were talking about, but with like cells from asymptomatic individuals to maybe compare that? Or like,
yes, so that is possible. And in fact, it may be something that will be necessitated in any case by the fact that luckily a lot of the infections now are more asymptomatic because also they're in younger individuals. So that's interesting to see what's the difference. It may be that it might become difficult to detect responses in the first place, in the sense that it's quite possible that asymptomatic individuals may have just generally a weaker infection and might induce a weaker immune response.
okay.
But that's not,
I don't know.
So I think it's worth looking.
In asymptomatic people, they still like produce enough virus, right, to like be able to infect other people.
Yes, and also they usually are seropositive, so for antibodies. So induce an immune response. There is some indication that the weaker the infection, the weaker the immune response, and it might be become undetectable faster. That is not necessarily foolproof, in the sense that you could make, so one argument you could say, okay, is the immune system is a system based on memory, so like your memory, if you almost got killed by being run over by a truck, you will remember that for a long time. I suppose if I ask you what you had for lunch 2 weeks ago, you may not remember anymore because it was not a very consequential event in your life.
That's a good point.
On the other hand, you could make the opposite argument. You could say people that have asymptomatic infection is because they have a much stronger, better, faster immune response. So basically those are the champions, right? And they are so good that they manage to have an asymptomatic infection. So it could be that it's a mixed bag. You can have some people with asymptomatic infection that just basically didn't have enough of an inoculum and they really didn't get infected very much and had such a mild infection. Or it could be that some people have, such a fantastic immune system, and those you would see a blazing hot immune response in an asymptomatic situation. Worth a look. Worth a look.
Okay, so if nobody else has any questions, I think it's 6 o'clock anyway, which is when the talk was supposed to end. So thank you, Dr. Sett, for speaking with us. This was a really enjoyable talk.
My pleasure. Thank you for having me.
Yep. Okay. Thank you, everyone who came to participate.
Cheers. Goodbye.
Goodbye.
Thank you for this amazing talk, Professor.
Thank you.
Thank you, Professor.